Basics of Medical Biochemistry. First edition, 2023. Chapter 12. Metabolism of Lipids. Part 2 of 3. By Professor Dr. Muhammad Jawad. MBBS, Tao, M. Philosophy, Ph.D., Dip M.E. Head of the Biochemistry Department-Fazaia Ruth Pfau Medical College Air University, PAF Base Faisal-Karachi, Pakistan. Ex-Professor of Biochemistry, Qassim University, Kingdom of Saudi Arabia. Ex-Professor, Head of the Biochemistry Department and Vice Principal. Liaquat College of Medicine and Dentistry and Fatima Jinnah Dental College, Karachi, Pakistan. Chapter 12. Metabolism of Lipids. 12.7 Ketone Bodies, KBS. Ketone bodies are a group of 3 water-soluble compounds containing the ketone group. They are produced by the liver from fatty acids during periods of starvation and other metabolic conditions. They include acetone, acetoacetate, and hydroxybutyrate. 2.7.1 Functions of ketone bodies. Ketone bodies, being water-soluble, are easily transported from the liver to various tissues. The 2 ketone bodies, acetoacetate and beta-hydroxybutyrate, serve as important sources of energy for peripheral tissues like skeletal muscle, cardiac muscle, renal cortex, etc. The production and utilization of KBS become more significant when glucose is in short supply to tissues, as observed in starvation and diabetes mellitus. During prolonged starvation, KBS are the major source of fuel for the brain. RBCs cannot utilize KBS due to the absence of mitochondria. Figure 12.22 Ketone bodies. 2.7.2 Ketogenesis. Ketogenesis is the synthesis of ketone bodies. Site: mitochondria of the liver. They are released from the liver to the blood for use as metabolic fuel by extrahepatic tissues. Ketone bodies are synthesized from acetyl-CoA. Acetyl-CoA is derived mainly from the beta-oxidation of fatty acids, and also from the catabolism of ketogenic amino acids, example leucine. Synthesis occurs under conditions of high rates of lipolysis as well as beta-oxidation of fatty acids, example long-term starvation or uncontrolled diabetes. High-fat/low carbohydrate diets, ketogenic diet. When oxaloacetate supplies are restricted and limited to utilizing all acetyl-CoA through the TCA cycle, the excess acetyl-CoA is converted to ketone bodies. Figure 12.23 Synthesis of ketone bodies, ketogenesis. Steps of ketogenesis. 1. Formation of acetoacetyl-CoA. The acetoacetyl-CoA is formed by the condensation of 2 acetyl-CoA molecules by the enzyme ketothiolase, also known as acetyl-CoA acetyltransferase. 2. Formation of beta-hydroxybeta-methylglutaryl-CoA, HMG-CoA. It is formed by the addition of another molecule of acetyl-CoA to acetoacetyl-CoA by the enzyme HMG-CoA synthase. 3. Formation of acetoacetate, a ketone body, by splitting of HMG-CoA. HMG-CoA lyase cleaves HMG-CoA to produce acetoacetate and acetyl-CoA. 4. Formation of remaining 2 ketone bodies, beta-hydroxybutyrate and acetone. Beta-hydroxybutyrate dehydrogenase converts acetoacetate to beta-hydroxybutyrate. Slow and spontaneous decarboxylation or by acetoacetate decarboxylase of acetoacetate also produces acetone. The odor of acetone may be detected in the breath of a person who has a high level of acetoacetate in the blood. Acetoacetate and beta-hydroxybutyrate are transported through the blood to their target tissues, where they are converted back to acetyl-CoA. In fact, ketone bodies are fatty acids that are transportable without requiring fatty acid-binding proteins such as serum albumin. 2.7.3 Ketolysis. It is the process of the breakdown of ketone bodies. Ketone bodies are mainly synthesized in the matrix of liver mitochondria, but these components cannot be utilized by the liver due to a deficiency of enzyme thiophorase. Ketone bodies oxidized in mitochondria of aerobic tissues such as muscle, heart, kidney, intestine, and brain. Beta-hydroxybutyrate is converted to acetoacetate by beta-hydroxybutyrate dehydrogenase. Acetoacetate, a chief ketone body, diffuses from the liver to the blood and is transported to peripheral tissues. In these tissues, acetoacetate is activated by the transfer of CoA molecules from succinyl CoA to form acetoacetyl CoA. This reaction is catalyzed by thiophorase. Figure 12.24 Ketolysis 12.7.4 Ketosis and ketoacidosis. Ketosis is a pathological condition that occurs when acetoacetate is produced faster than its catabolism and utilization. It is the overproduction of ketone bodies that causes ketonemia and ketonuria. Ketonemia is higher than normal blood level of KBS that occurs during excessive synthesis of KBS by the liver more than the catabolic capacity of the peripheral tissues. The concentration of total ketone bodies in blood at the well-fed stage is 1-3 mg/dl. Ketonuria is the increase of output of KBS in urine, normally 1 mg per day. Ketosis is the overall accumulation of ketone bodies in the blood, ketonemia, and urine, ketonuria. Ketoacidosis, excessive buildup of ketone bodies results in ketosis, eventually leading to a fall in blood pH due to the acidic ketone bodies. Diabetic ketoacidosis, DKA. It is a state of inadequate insulin levels resulting in high blood sugar and accumulation of organic acids and ketones in the blood. It is also common in DKA to have severe dehydration and significant alterations in the body's blood chemistry. Essential diagnostic features of diabetic ketoacidosis are hyperglycemia greater than 250 mg/dl, acidosis with blood pH 7.3, serum bicarbonate 15 milliequivalent/L, and serum positive for ketone bodies. The main signs and symptoms are polyuria, polydipsia, fatigue, nausea and vomiting, mental stupor leading to coma, fruity breath, acetone, hypotension with tachycardia, hypothermia, abdominal pain, and tenderness. Ketoacidosis in starvation. Prolonged fasting may result from an inability to obtain food or the desire to lose weight rapidly, or in clinical situations in which an individual cannot eat because of trauma, surgery, neoplasms, burns, etc. The plasma levels of glucose, amino acids, and triacylglycerols fall. These changes trigger a decline in insulin secretion and an increase in glucagon release. The decreased insulin to glucagon ratio makes this period of nutritional deprivation a catabolic state, characterized by the degradation of glycogen, triacylglycerol, and protein. This sets into motion an exchange of substrates between the liver, adipose tissue, muscle, and brain that is guided by the following two priorities. The need to maintain glucose levels to sustain the energy metabolism of the brain, red blood cells and other glucose-requiring cells. 2. To supply energy to other tissues by mobilizing fatty acids from adipose tissue and converting them to ketone bodies to supply energy to other cells of the body. 12.8 Metabolism of cholesterol. Cholesterol is a derived lipid, a waxy steroid found in the cell membranes and transported in the blood plasma of all animals. Its structure is characterized by the presence of a steroid nucleus known as per-hydrocyclopentanophenonthrene. It is an essential component of mammalian cell membranes where it is required to establish proper membrane permeability and fluidity. It is widely distributed in all cells of the body, particularly in nervous tissue, muscles, skin, liver, intestine, and heart. Cholesterol is the principal sterol synthesized by animals, but small quantities are synthesized in other eukaryotes, such as plants and fungi. It is almost completely absent among prokaryotes, which include bacteria. Cholesterol level in the body is maintained by keeping a balance between exogenous supplies from diet and de novo synthesis mainly in the liver. 12.8.1 Biosynthesis of cholesterol. Cholesterol is synthesized by using acetyl-CoA molecules as a precursor. Its biosynthesis mainly takes place in the liver, but other tissues like the skin, intestine, adrenal cortex, aorta, gonads, etc. are also involved in its synthesis. The liver synthesizes 50 to 60% of the total endogenous cholesterol of the body, whereas 15 to 20% is synthesized in the intestine. There are 4 major steps involved in its biosynthesis. 1. Formation of mevalonic acid. mevalonate. Its formation occurs through the following three substeps. I. Formation of acetoacetyl CoA. Two acetyl CoA molecules are condensed to give acetoacetyl CoA by the enzyme ketothiolase, acetyl CoA acetyltransferase. 2. Formation of beta-hydroxy-beta-methylglycerol CoA. HMG-CoA acetoacetyl CoA condenses with another acetyl CoA molecule to form beta-hydroxy-beta-methylglycerol CoA. The reaction is catalyzed by HMG-CoA synthase. HMG-CoA can also be written as 3-hydroxy-3-methylglycerol CoA. A free CoA molecule is released in both of the above steps. 3. Reduction of HMG-CoA to mevalonate. HMG-CoA is reduced to mevalonic acid, 6C compound. This reaction requires 2-NADPH and it is catalyzed by HMG-CoA reductase, which is the regulatory enzyme. This reaction is the rate-limiting step of cholesterol biosynthesis, and the HMG-CoA reductase enzyme is the rate-limiting enzyme. 2. Synthesis of cholesterol from mevalonate. It involves the following reaction. I. By successive addition of 2-ADP, mevalonate is converted to 5-pyrophosphomevalonate, 6C compound. The reaction is catalyzed by mevalonate kinase. 2. The 5-pyrophosphomevalonate undergoes decarboxylation and dephosphorylation reactions by the action of enzymes mevalonate decarboxylase and mevalonate phosphatase respectively, as a result of which a 5-carbon containing compound isopentenyl pyrophosphate, IPP, is formed. Figure 12.25 Formation of mevalonate. 3. One molecule of isopentenyl pyrophosphate is epimerized into 3,3-dimethyl-allyl pyrophosphate, DPP, by the action of the enzyme isopentenyl pyrophosphate isomerase. These two 5-carbon containing compounds, IPP and DPP, are collectively known as isoprenoid units. 4. In the presence of the enzyme geranyl pyrophosphate synthase, geranyl dimethyl-allyl transferase, the isoprenoid units, IPP and DPP, are condensed together to form a 10-carbon compound known as geranyl pyrophosphate. A molecule of pyrophosphate is released during this reaction. Geranyl pyrophosphate is then condensed with another molecule of isopentenyl pyrophosphate to form a 15-carbon compound known as farnesyl pyrophosphate by the action of enzyme farnesyl pyrophosphate synthase. One molecule of pyrophosphate is released during this reaction. 6. Two molecules of a 15-carbon compound, i.e., farnesyl pyrophosphate, condensed together to form a 30-carbon compound, i.e., squalene. This reaction is catalyzed by the action of the enzyme squalene synthase in the presence of NADPH plus H+. One molecule of pyrophosphate is also released during this reaction. 7. Squalene is now converted to linosterol. First, squalene is acted upon by the enzyme monoxygenase in the presence of O2 and NADPH plus H+ to become squalene epoxide. Then enzyme squalene synthase causes the cyclization of squalene epoxide and forms linosterol, the sterol. 8. Linosterol is finally converted to cholesterol. This conversion involves the following changes. Removal of methyl groups as CO2 in the presence of NADP plus H+, NAD+, and O2. Reduction of double bonds. Shifting of double bonds. Very little cholesterol is excreted as most are recycled back to the liver. The cholesterol that is excreted is first converted to water-soluble bile acids or bile salts. Cholesterol is the main component of gallstones, and it was from a gallstone that cholesterol was first isolated. Figure 12.26 Synthesis of cholesterol from mevalonate, P, phosphate, PP, pyrophosphate. 12.8.2 Regulation of cholesterol synthesis. The process of cholesterol biosynthesis in the liver is strongly regulated by dietary cholesterol levels. When dietary intake of cholesterol is high, synthesis is decreased, and when dietary intake is low, synthesis is increased. Cholesterol and similar sterols, bile acids and bile salts, act as regulatory molecules to maintain healthy levels of cholesterol. Cholesterol biosynthesis is regulated by the following mechanisms. 1. Regulation through feedback inhibition. the enzyme HMG-CoA reductase in the liver is inhibited by cholesterol formed at the end of the cholesterol biosynthetic pathway or by dietary cholesterol. When HMG-CoA reductase is inhibited, the remaining steps in the pathway are stopped. Sterol regulatory element binding proteins, SREBPs, are transcription factors that bind to the sterol regulatory element of DNA. SREBPs are encoded by the genes SREBP1 and SREBP2 genes, required for de novo lipogenesis. SREBP2 regulates the genes of cholesterol metabolism. It becomes active when the intracellular cholesterol level falls. SREB cleavage activating protein, SCAP, is a potent activator of SREB pathways in cholesterol synthesis. In the presence of high cellular sterol levels, SCAP keeps SREB within the endoplasmic reticulum. When cholesterol concentration is low, SCAP guides SREB from the endoplasmic reticulum to the Golgi apparatus, where SREB undergoes two proteolytic cleavage steps to release the mature and functionally active transcription factor called nuclear SREB, NSREBP. The NSREBP translocates to the nucleus and binds to sterol response elements, SRE, and activates the target genes. 2. Regulation by covalent modification. The enzyme HMG-CoA reductase is also regulated by covalent modification through phosphorylation and dephosphorylation. Phosphorylation of HMG-CoA reductase by the kinase in the presence of ADP in the cytosol of the liver inactivates the enzyme. On the other hand, dephosphorylation of HMG-CoA reductase by the enzyme phosphatase reactivates it. 3. Hormonal regulation. Some hormones are also involved in the regulation of cholesterol biosynthesis. They tend to control the activity of HMG-CoA reductase either by influencing its synthesis or by its covalent modification. Insulin increases HMG-CoA reductase activity, whereas glucagon and cortisol decrease their activity. 4. Pharmacological inhibitors of HMG-CoA reductase. The statins, a group of cholesterol-lowering drugs, have been effective and used to control cholesterol in the blood. They include atorvastatin, fluvastatin, rosuvastatin, etc. They act as competitive inhibitors of HMG-CoA reductase. 12.8.3 Transport of cholesterol. Cholesterol is transported in the blood as part of soluble complexes called lipoproteins. Following are the main types of lipoproteins involved in the transportation of cholesterol. I. Chylomicron. It is a fat droplet containing little protein. It picks up dietary cholesterol from the intestine. Fats are delivered to adipose tissue, leaving a chylomicron remnant containing mostly cholesterol that is brought into the liver by binding with receptors that recognize ApoE in the chylomicron remnant. 2. Very low density lipoprotein, VLDL. It is synthesized in the liver and contains excess triacylglycerol and cholesterol. It transports cholesterol from the liver to extrahepatic tissues. During transport in the bloodstream, VLDL delivers triacylglycerol to tissues, leaving IDL molecules. The IDL molecules lose triacylglycerols in the bloodstream until they form LDL molecules, which have the highest percentage of cholesterol within them, thus LDL is called bad cholesterol. 3. Low-density lipoprotein, LDL. It is taken up by peripheral tissues through LDL receptors which recognize ApoB100. Cholesterol is liberated and stored as a cholesterol ester that is produced by the action of acyl-CoA cholesterol acyltransferase, ACAT. 4. High-density lipoprotein, HDL. It removes cholesterol from extrahepatic tissues and esteraphing it using plasma enzyme lecithin cholesterol acyltransferase, LCAT, thus HDL is called as good cholesterol. HDL is taken up by the liver after binding with HDL receptors which recognize ApoA1 and hydrolyzed to liberate cholesterol. Released cholesterol either enters the structure of other lipoproteins or bile salts or is excreted in bile. 12.8.4 Regulation of blood cholesterol level. The body gets cholesterol both exogenously from the diet and endogenously by synthesizing within the cells, mainly in the liver. The body synthesizes 1.5 to 2.0 g of cholesterol every day from acetyl-CoA units, and the average daily dietary intake is 0.4 to 0.5 g. The normal blood cholesterol level varies with age, sex, diet, and geographic or cultural region. However, an acceptable fasting level in an adult is 140-199 mg/dl, given by most healthcare authorities. Table 12.2 Conditions associated with elevated and decreased blood cholesterol levels. Elevated cholesterol levels. Decreased cholesterol levels. Type 2 familial hypercholesterolemia. Hyperlipoproteinemias type I, 4 and V. Cholestasis. Hepatocellular disease. Nephrotic syndrome, glomerulonephritis. Chronic renal failure. Alcoholism. A diet high in fat and cholesterol. Obesity. Poorly controlled diabetes mellitus. Hypolipoproteinemia. Myeloproliferative disease. Malabsorption syndrome. Severe burns, inflammation. Chronic obstructive pulmonary diseases. COPD. Hypercholesterolemia is associated with atherosclerosis of vital arteries, causing cerebrovascular, coronary and peripheral vascular disease by narrowing and hardening of the arteries. Heart patients are strongly advised to take all care to bring their blood cholesterol level near 150 mg/dl. Blood cholesterol level increases progressively with age but again decreases in very old people. Socioeconomic conditions. Generally rich and well-off populations, particularly those with a sedentary lifestyle, have higher blood cholesterol levels. Diet. Foods derived from animals such as egg yolks, meat, poultry, fish, seafood, and whole milk dairy products contain high amounts of cholesterol. Foods derived from plants such as fruits, vegetables, grains, nuts, and seeds do not contain cholesterol. It is the recommendation of various health agencies working on the prevention of heart diseases, to limit average daily cholesterol intake to less than 300 mg. In case of the presence of heart disease, it is further dropped to less than 200 mg daily. Food high in saturated fat should be avoided as it generally contains large amounts of dietary cholesterol. Exercise. Regular physical activity can significantly help to reduce LDL, i.e., bad cholesterol, and raise HDL, i.e., good cholesterol levels. Intake of alcohol, red wine, in moderate amounts tends to increase HDL. Regular physical activity of 150 minutes a week or 30 minutes daily for 5 days a week is required to keep healthy. It also helps to lose weight. Weight, body mass index, BMI, greater than 25 and waist circumference greater than 40 inches indicate high blood cholesterol levels. Table 12.3 Factors affecting plasma cholesterol levels. Factors increasing plasma cholesterol levels. Factors decreasing plasma cholesterol levels. 1. Increased plasma FFA levels, any condition increasing plasma FFA level causes increased secretion of hepatic VLDL. It is seen in diabetes mellitus, smoking, and emotional stresses. 2. Hypothyroidism. The physiological level of thyroid hormone, TH, is essential for maintaining cholesterol homeostasis. Patients with hypothyroidism typically have elevated plasma cholesterol and increased lipid accumulation in the liver. 3. Diet rich in saturated fats and cholesterol. 4. Familial and genetic predisposition. 5. Nephrotic syndrome. 6. Sedentary lifestyle. 1. Low caloric intake, undernourished. Conditions are associated with increased production of ketone bodies and low cholesterol synthesis. 2. High intake of polyunsaturated fatty acids. It causes increased loss of cholesterol in the intestine as bile acids. 3. Hyperthyroidism. 4. Estrogen. Estrogen plays an important role in the modulation of the total cholesterol level by reducing LDL and concurrently increasing HDL. 5. Drugs. The statins, like atorvastatin, fluvastatin, rosuvastatin, etc. act as competitive inhibitors of HMG-CoA reductase. Clofibrate is used for Controlling high cholesterol and triacylglycerol levels in the blood. It increases lipoprotein lipase. Activity to promote the conversion of VLDL to LDL, thus reducing VLDL levels. Niacin. Nicotinic acid is also used to lower cholesterol levels. 12.8.5 Fate, utilization of cholesterol. Cholesterol after being synthesized in the body serves as a precursor of some very important molecules such as vitamin D, bile acids, sex hormones, glucocorticoids, mineralocorticoids. 12.8.6 Degradation of cholesterol. Very little cholesterol is excreted as most are recycled back to the liver. It is usually excreted as water-soluble bile acids or bile salts. A small amount of cholesterol is acted upon by bacteria in the intestine to its isomers such as coprostanol and cholestanol. These compounds are removed from the body through feces as neutral fecal sterols. 12.9 Metabolism of lipoproteins. Lipoproteins are complex particles that have a central hydrophobic core of nonpolar lipids, primarily cholesterol esters and triacylglycerols, tags. This hydrophobic core is encircled by a hydrophilic membrane consisting of phospholipids, free cholesterol, and apolipoproteins. 12.9.1 Metabolism of chylomicrons. 1 Nascent chylomicrons are assembled in intestinal mucosal cells. They are rich in TAG produced from dietary lipids. They enter the lymphatic system and then enter the blood circulation via the thoracic duct. 2 Nascent chylomicrons contain apoprotein B48 but pick up other apoproteins, ApoC2 and ApoE, from HDL once they enter the circulation. Their function is to transport dietary lipids to the adipose tissue and skeletal muscles. 3 The TAG components of chylomicron are hydrolyzed by the activity of the lipoprotein lipase, allowing the released free fatty acids to be stored in adipose tissue or used for energy in muscle. Glycerol is also released, which is mostly transported to the liver. Lipoprotein lipase is an extracellular enzyme activated by ApoC2. 4 ApoC2 is separated and returned to HDL, and chylomicron becomes chylomicron remnant, which is rich in cholesterol ester. 5 Chylomicron remnant is picked by the liver through ApoE-specific receptors. Inside the hepatocytes, the endocytosed vesicles are fused with lysosome and their contents are hydrolytically degraded. Figure 12.27 Structure of a chylomicron. Figure 12.28 Metabolism of chylomicron. Source: www.medicalpps.org. 12.9.2 Metabolism of very low-density lipoprotein, VLDL. 1. These are synthesized in the liver. Contain mostly endogenously synthesized TAG but with a significant amount of cholesterol and cholesterol ester. 2. VLDLs travel around the circulatory system until they associate with lipoprotein lipase, an enzyme bound to the endothelial surface. Nascent VLDL contains ApoB100 but picks up others, ApoC2 and ApoE, from HDL in circulation. 3. The TAG components of chylomicron are hydrolyzed by the lipoprotein lipase, allowing the released free fatty acids to be stored in adipose tissue or used for energy in muscle. 4. ApoC2 and ApoE are separated and returned to HDL, and triacylglycerol is lost. The VLDL shrinks and forms IDL and LDL, which are rich in cholesterol ester and cholesterol. 5. LDL is taken up by the liver or extrahepatic tissues and is endocytosed. The fate of lipoproteins remnant. The liver is responsible for the uptake of lipoprotein remnants. Chylomicron remnants are taken up by receptor-mediated endocytosis. VLDL is converted to intermediate density lipoprotein, IDL, and eventually to LDL. VLDL IDL, VLDL remnant, LDL. The fate of IDL, VLDL remnant, is either taken by the liver directly via LDL receptor, needs ApoB100, ApoE, or it is converted to LDL. Figure 12.29 Metabolism of very low-density lipoprotein, VLDL, and low-density lipoprotein, LDL. Source: www.medicalpps.org. 12.9.3 Metabolism of low-density lipoprotein, LDL. LDL is metabolized via the LDL receptor, ApoB100, ApoE. It is formed from circulating VLDL either directly or via IDL by the loss of triacylglycerol from them. 30% of LDL is degraded in extrahepatic tissues and 70% in the liver. 1 LDL bind to specific cell receptors located on the plasma membrane of target cells. 2. The LDL receptor is a glycoprotein that contains a domain with negatively charged residues. 3. This LDL binding domain has electrostatic interactions with the positively charged arginine and lysine residues of ApoB100. 4. The LDL receptors migrate to areas of the plasma membrane specialized for endocytosis called coated pits. They are called coated pits because of the clathrin protein. Table 12.4 Plasma levels of LDL cholesterol, LDL-C. Values in mg/dl. Increased in optimal 100. Near optimal equals 100 to 129. Borderline high equals 13 to 159. High equals 160 to 189. Very high greater than 190. Familial type 2 hyperlipidemia. High saturated fat and high cholesterol diet. Nephrotic syndrome. Diabetes mellitus. Nephrotic syndrome. Chronic renal failure, CRF. Hyperlipidemia secondary to hypothyroidism. Anorexia nervosa. Porphyria. Decreased in hypolipoproteinemia. Type I hyperlipidemia. ApoC2 deficiency. Chronic anemias. Hyperthyroidism. Acute stress, burns, illness. Inflammatory joint disease. Estimation of LDL cholesterol by Friedewald formula. The plasma value of LDL-C can be calculated by the Friedewald formula, provided that other plasma lipid fractions are known. It is valid only if the specimen is obtained in fasting condition and Tg values must be 400 mg/dl. Figure 12.30 Friedewald formula, LDL-C, LDL cholesterol, HDL-C, HDL cholesterol, Tc, total cholesterol, Tg, triglycerides. Note, all values are in mg/dl. Example 1. The lipid profile of a 50-year-old male showed Tc 290 mg/dl, Tg 370 mg/dl and HDL-C 29 mg/dl. Calculate the LDL-C of this patient. LDL-C equals Tc, Tg, HDL-C. 5. LDL-C equals 290 to 370, 29. 5. LDL-C equals 290 74, 29. LDL-C equals 216 to 29. LDL-C equals 187 mg/dl. Example 2. The lipid profile of a 45-year-old male showed Tc 250 mg/dl, Tg 350 mg/dl and HDL-C 28 mg/dl. Calculate the LDL-C of this patient. LDL-C equals Tc, Tg, HDL-C. 5. LDL-C equals 250 to 350, 28. 5. LDL-C equals 250 70, 28. LDL-C equals 280 to 28. LDL-C equals 152 mg/dl. 12.9.4 Metabolism of high-density lipoproteins, HDL. 1 HDL is synthesized and secreted from both the liver and intestine. It acts as a repository for ApoC and ApoE, which are required for the metabolism of chylomicrons and VLDL. Normal levels of HDL in male is 35-65 mg/dl and in female is 35-80 mg/dl. 2 Nascent HDL consists of discoid phospholipids bilayer containing ApoA and free cholesterol. Role of lecithin cholesterol acyltransferase, LCAT. LCAT is synthesized by the liver. It binds to nascent HDL and is activated by ApoAI. LCAT transfers the fatty acid from carbon 2 of phosphatidylcholine to cholesterol. This produces a hydrophobic cholesterol ester, which is sequestered in the core of the HDL, and lysophosphatidylcholine, lysolecithin, which binds to albumin. Lecithin plus cholesterol, lysolecithin plus cholesterol ester. ApoAI activates LCAT and it is also the ligand for the HDL receptor. ApoA2 inhibits LCAT enzyme. 3. The cholesterol efflux is brought about by the esterification of cholesterol under the effect of LCAT. The cholesterol ester-rich HDL, HDL2, gains entry through the scavenger receptor, SRB1. Figure 12.31 Action of lecithin cholesterol acyltransferase, LCAT, enzyme. HDL cycle. The discoidal nascent HDL is converted to HDL3 after getting cholesterol ester from free cholesterol by the action of LCAT. HDL3 accepts more cholesterol from the tissues and the cholesterol is then esterified by LCAT, increasing the size of the particles to form the less dense HDL2. HDL3 is again reformed, either after selective delivery of cholesterol ester to the liver via the SRB1 or by hydrolysis of HDL2 phospholipid and triacylglycerol by hepatic lipase. This interconversion of HDL2 and HDL3 is known as the HDL cycle. Free ApoAI is converted to pre-HDL after associating with the required amount of phospholipid and cholesterol. The release of cholesterol from peripheral cells is mediated, at least in part, by the transport protein, ADP binding cassette transporter A1, ABCA1. The cholesterol esters are taken up by the liver through its cell surface receptor, SRB1, scavenger receptor class B type 1, that binds HDL. It is to be noted that the hepatic lipase, with its ability to degrade both TAG and phospholipids, also participates in the conversion of HDL2 to HDL3. Functions of HDL. 1. HDL is a reservoir of apolipoproteins, ApoC2 and ApoE. 2. HDL uptake of unesterified cholesterol. They take up cholesterol from non-hepatic, peripheral, tissues and return it to the liver as cholesterol esters. 3. Esterification of cholesterol. When cholesterol is taken up by HDL, it is immediately esterified by the plasma enzyme LCAT. As the discoidal nascent HDL accumulates cholesterol esters, it first becomes a spherical, relatively cholesterol ester-poor HDL3 and, eventually, a cholesterol ester-rich HDL2 particle that carries these esters to the liver. Cholesterol ester transfer protein, CETP, moves some of the cholesterol esters from HDL to VLDL in exchange for triacylglycerol. 4. Reverse cholesterol transport. The selective transfer of cholesterol from peripheral cells to HDL, and from HDL to the liver for bile acid synthesis or disposal via the bile, and to steroidogenic cells for hormone synthesis. Figure 12.32 Metabolism of high-density lipoprotein, HDL, FC, free cholesterol, CE, cholesterol ester, PL, phospholipid, LCAT, lecithin cholesterol acyltransferase, CETP, cholesterol ester transfer protein, ABCA1, ADP binding cassette transport protein, SRB1, scavenger receptor class B type 1, source, www.semantic.com. 12.9.5 The total cholesterol/HDL cholesterol ratio. The total cholesterol/HDL ratio helps to estimate the risk of developing atherosclerosis. High ratios indicate a higher risk of heart attacks, whereas low ratios indicate lower risk. Table 12.5 Interpretation of the total cholesterol/HDL cholesterol ratio. Average risk. 2 times average risk. 3 times average risk. Male. 5.0. 9.6. 23.4. Female. 4.4. 7.1. 11.0. 12.9.6 LDL/HDL ratio. the LDL/HDL ratio is a purer ratio than total cholesterol/HDL because LDL is a measure of bad cholesterol and HDL is a measure of good cholesterol. The LDL/HDL cholesterol ratio is more important than individual levels of HDL cholesterol and LDL cholesterol. However, a recent Heart Foundation recommendation is that irrespective of the ratio, one should try to reduce LDL cholesterol. Table 12.6 Interpretation of the LDL/HDL cholesterol ratio. Risk level: low risk, average risk, moderate risk, high risk. LDL/HDL ratio: 3.3, 4.4, 4.4, 7.1, 7.1, 11.0, 11.0. 12.10 Bile acids and bile salts. The end product of cholesterol utilization is the bile acids. Bile acids are polar derivatives of cholesterol. Synthesis of bile acids is one of the predominant mechanisms for the excretion of excess cholesterol. These compounds are synthesized in the liver, stored in the gallbladder and then released into the small intestine. The most abundant bile acids in human bile are cholic acid and chenodeoxycholic acid, which are identified as primary bile acids. The salts of bile acids with glycine and taurine are called bile salts. They are secreted into the gallbladder through the bile duct. They are the major constituents of bile and solubilize dietary lipids. Bile salts, by reducing the surface tension, promote the hydrolysis of lipids by the enzyme lipase and also facilitate the absorption of lipids through the process of emulsification. Figure 12.33 Structure of primary bile acids. 12.10.1 Synthesis of bile acids. Cholesterol is converted to 3,7,12-trihydroxycholinic acid, i.e., cholic acid, after hydroxylation at C3, C7, and C12 and reduction at C5 and carboxylation at C26. Enzymes involved in these reactions are mixed function oxidase, reductase, and carboxylase respectively. Cholic acid is then converted to cholyl-CoA by the action of the enzyme cholyl-CoA synthetase. A molecule of CoA is required for this reaction. The activated carboxyl carbon of cholyl-CoA is then condensed with either amino group of glycine or taurine to form glycocholic acid or taurocolic acid respectively. Secondary bile acids are produced from primary bile acids by the action of intestinal flora, which causes the removal of glycine and taurine along with dehydroxylation. Deoxycholic acid and lithocholic acid are secondary bile acids derived from cholic acid and chenodeoxycholic acid respectively. Figure 12.34 Synthesis of bile acids. Note: synthesis of secondary bile acids takes place in the intestine by microbial enzymes. In the gut, after the removal of glycine and taurine residues, bile acids are either excreted, small percentage, or reabsorbed by the gut and returned to the liver. A major amount is reabsorbed and only a small fraction of bile salts are excreted in feces per day. 12.10.2 Enterohepatic circulation of bile salts. A large amount, 90 to 95%, of bile salts that enter to duodenum are absorbed back into the portal vein by apical sodium bile salt cotransporter and returned to the liver. This is known as the enterohepatic circulation of bile salts. The remaining 5 to 10% enter the colon, acted upon by colonic bacteria to form secondary bile salts, i.e., deoxycholic acid and lithocholic acid. Daily about 15 to 30 grams of bile salts are secreted by the liver into the duodenum. But only about 0.5 grams of bile salt is lost in feces. Clinical aspects. Cholestyramine, a bile acid sequestrant, can be used in the treatment of hypercholesterolemia. It binds with bile salts in the intestine and prevents their reabsorption, thus enhance their excretion. Dietary fibers also have the same action. Figure 12.35 Enterohepatic circulation of bile salts. Source: ResearchGate. Exercise questions. For MCQ 1 to 5 see Part 1 and for MCQ 11 to 15 see Part 3 of this chapter. MCQ 6. A 58-year-old obese man consulted a physician for complaints of weakness, easy fatigability, joint pain, and shortness of breath during physical activity. Complete checkup and lab investigations revealed that he is suffering from hyperlipoproteinemia type IIA. Which of the following lipoproteins is most likely elevated in this patient? Chylomicron, HDL, IDL, LDL, VLDL. MCQ 7. Elevated levels of chylomicron were measured in the blood of patients. Which of the following dietary reduction would be most helpful in lowering chylomicron levels? Cholesterol, fat, starch, sugar. Total calories. MCQ 8. A patient suffering from dyslipidemia was specifically diagnosed with one of the sphingolipidoses based on symptoms and lab investigation. Which of the following chemical structures is found in the compound that is accumulated in this patient? A glycerol backbone, a steroid ring with sphingosine, ceramide, N-acetylneurominic acid, NANA, phosphorylcholine. MCQ 9. Which of the following enzymes is increased via the rate of transcription of the respective mRNA in an individual who is on a fat-free, high-carbohydrate diet? A. Fatty acid synthase, B. Fructose 1,6-bisphosphatase, C. Hormone-sensitive lipase, D. Phosphenol pyruvate carboxykinase, E. Pyruvate carboxylase. MCQ 10. Which of the following enzymes is involved in the initial step during the synthesis of triglycerides from free fatty acids and glycerol in the liver? Glycerol kinase, glycerol 3-phosphate dehydrogenase, glyceraldehyde 3-phosphate dehydrogenase, lipoprotein lipase, hormone-sensitive lipase. End of Part 2 of 3 of Chapter 12. Thank you.